Last time we talked about acids, bases, pH, and organic compounds. In today's video, I will talk about proteins, DNA, RNA, types of molecules, DNA replication, and ATP. Don't forget to take notes if necessary. Let's begin. Proteins are made up of amino acids. Examples of amino acids are alanine, valine, glutamic acid, tryptophan, tyrosine, and histidine. Amino acids contain atoms of carbon, hydrogen, oxygen, nitrogen, and sometimes contain sulfur and phosphorus. There are 20 different types of amino acids, each having an amino group, a carboxyl group, and a radical residue attached. Proteins are catalysts of metabolic reactions. They deal with the transport of oxygen and have a structural role. Examples of proteins are enzymes, insulin, the growth hormone, hemoglobin, keratin, and collagen. Small proteins are called peptides. The formation of a protein occurs when amino acids bind to each other by removing the hydrogen atom from the amino group of one amino acid and removing the hydroxide from the acid group of the second amino acid. Then, the amino group of the first amino acid binds to the acid group from the second amino acid and a water molecule is removed. Therefore, a dehydration reaction takes place. The bond formed is called a peptide bond. Hormones are proteins that deal with the chemical regulation of the body. Enzymes catalyze most of the chemical reactions that take place in the cells. They are not consumed in the reaction in the cells. Therefore, they remain available to catalyze future reactions. Without enzymes, cellular chemistry cannot take place. Outside the cell, proteins are found in bone cartilage, tendons, and ligaments. Information about protein synthesis is found in the nucleus of cells, where the genetic code is. Amino acids can be used as energy when the liver removes the amino group of an amino acid and the body cells use the resulting compound for energy. Nucleic acids. Nucleic acids are large molecules made up of subunits called nucleotides. Each nucleotide contains a carbohydrate molecule linked to a phosphate group and a nitrogenous base molecule. There are two types of nucleic acids: DNA, the deoxyribonucleic acid, and RNA, the ribonucleic acid. DNA. In 1953, James Watson and Francis Crick proposed a universal model for the structure of DNA. It consists of two chains of nucleotides with the bases inwards, like the steps of a ladder. DNA is shaped like a double helix and is the stuff that genes are made of. DNA is found in the 46 chromosomes of the human cell. This acid contains a carbohydrate with 5 carbon atoms, which is a pentose, called deoxyribose, and contains the nitrogenous bases thymine, adenine, cytosine, and guanine. DNA is composed of phosphate groups that link sugars together and dictate the correct order of amino acids in the protein to be synthesized. The functions of DNA are protein synthesis and transmission of genetic information. RNA. It is found in the nucleolus, the nucleus, and the cytoplasm of the cell. It contains ribose, which is a pentose, which is a 5-carbon carbohydrate, and also contains phosphate groups that link the sugars together. Unlike DNA, RNA contains the nitrogen base called uracil, not thymine. RNA is found in the ribosomes and in the cytoplasm of the cell as messenger RNA or transfer RNA. The function of RNA is protein synthesis. Both DNA and RNA contain phosphate groups. Phosphate groups link the deoxyribose or ribose molecules to each other in the nucleotide chain to form the so-called backbone of the chain. Now I suggest you pause this video and screenshot this slide showing a comparison between DNA and RNA. Types of molecules. Adenine and guanine are purine molecules. Thymine, cytosine, and uracil are pyrimidine molecules. The bases of nucleotide chains are held together by weak hydrogen bonds. Adenine binds to thymine through a double bond. Cytosine is linked to guanine by a triple bond. Obviously, triple bonds are more resistant than double bonds. The sequence of DNA bases is the essence of the genetic code. DNA replication. DNA replication begins when specialized enzymes break or unwind the double helix. In human cells, 46 chromosomes are replicated. The replicated chromosomes separate, and 46 chromosomes will pass into each new cell. As the two strands separate, the enzyme DNA polymerase joins the nucleotide components to form a long string of nucleotides. Thus, each old DNA strand causes the synthesis of a new DNA strand by pairing complementary bases. DNA replication is done according to the semi-conservative model, that is, one old strand is preserved in each new DNA double helix. Next, we will talk about ATP. Adenosine triphosphate is a nucleotide in RNA that results from the energy of food molecules through cellular respiration and releases a phosphate group. I know it's a rather quick ending, but with that being said, we've come to the end of this video. Don't forget to press the like, share, and subscribe button. If you have any questions, please contact me on Instagram or Facebook. Please tell me what you thought about this video in the comments. Have a nice week.